Hey friends, Lob helps developers extend their applications into the physical world with automated direct mail delivered through RESTful APIs. Some notable features are sending postcards, letters, and checks as easily as email. You can use customizable HTML templates, verification to ensure deliverability, and autocomplete address forms. Listeners can receive a $20 account credit at lob.com/special-offers/Hansel That's lob.com/special-offers/Hansel or click on the logo below. Hi, this is Scott Hansel, and this is another episode of Hansel Minutes. Today I'm talking with Dr. Shawn Carbonell. He's the co-founder and managing partner of Brazen Capital, Brazen Bio, co-founder president of Cure Glioblastoma, and an Oxford-educated MD PhD. How are you, sir?
Very good. Thanks so much for having me.
So that's a very impressive intro and a very impressive LinkedIn. But of course, where did I find you? I found you on TikTok because that's where everybody is at. Exactly. Why did you get on TikTok? It seems like you have enough stuff to do that you would maybe not spend your time there.
It started off with me sort of trying to find a way to teach science to people on the internet. And originally I started a blog like 10 years ago, but I could never keep up. And so what I started doing was microblogging on Instagram, did that for 6 or 7 years, built up a community focused around startups and biotech, as well as science and brain cancers, which is what my nonprofit focuses on. And then someone from TikTok actually reached out to me to recruit me to their accelerator program, which is, they have multiple accelerators for different topics, Latinx. And the one I got recruited to was called Learn on TikTok. So it's about educational content.
Mm-hmm. Okay, wow. So someone said, we need you on our platform and we're going to make you, give you the tools to make you more successful.
Essentially, yes. And so there's office hours a couple times a month and we get free swag, obviously.
Oh, wow.
It's been great.
Did that feel weird? I feel weird as being a person of a certain age on TikTok. And I tried to figure out what my niche was. And I know that there's like old people dancing, but I decided that wasn't my thing. You know, my thing is about technology education and I do it in my style. How did you find your style and your voice?
It was really, I give a lot of credit to TikTok. I'm really grateful for them reaching out because I'm video shy and I'm an introvert and doing video was never a forte of mine. I never wanted to do it. One of my informal mentors is Gary V, as well as for a lot of people. But during the beginning parts of the pandemic, last year, Gary V was saying a lot about just get on TikTok, just point the camera at yourself and start talking, post it to Instagram stories. So I started doing that on Instagram stories, got more comfortable with that. And it just dovetailed into this time the agent reached out to me and said, we'd like you to make some content for us on TikTok. Basically, it's not my native medium, but I'm a creative person and, I found my way through it. And, my first hundreds or so videos are not very good, but I've eventually found my stride. So it's, it's learning from zero essentially and growing so.
That really resonates. I love that you said my first 100 videos because this podcast has been on for over 800 episodes and the first couple hundred were kind of mediocre, and people don't say stuff like that. People usually say, yeah, the first 3 or 4 were not very good, but then I found my stride. But no, people need to understand that, the first 10 years of blogging, it was kind of average. The last 10 have been pretty awesome. It takes practice. A while. Overnight success in 20 years, right?
Exactly.
Have you found though that I like TikTok. I find it very positive. I find it to be a very welcoming community until the bad guys find you. And the keyboard warriors, they're just like, it's the user with like 9 digits at the end of their name people, or people with a frog as their avatar or whatever. What's been the ratio of positivity versus trolling that you've seen?
It's funny you say the word ratio because that does sometimes happen on TikTok, not just a Twitter thing. So it really depends. Sometimes obviously I'm speaking about the vaccine and boosters, and sometimes it starts off on the right side of TikTok and I have a lot of followers sort of blowing up my video. Last night, it started off sort of on the wrong side, which also has the same effect. And eventually it sort of evens out. So, my most viral video was 16.6 million views. That was the one in July, my 4-month data after the Pfizer vaccine. And there are over 20,000 comments. And I think the majority are negative. So
really,
it's surprising, but at the same time, it's sort of why things go viral, and I just hope that the right people see it.
Yeah, yeah, and I think that as a person with that many followers and that many comments, you would agree that the commenting and triaging of comment system in TikTok is trash. There's just no way to keep up. You see a comment if it happens to be the first 100, but otherwise it's just infinite scrolling. It's difficult.
Yeah, I keep reminding my community manager at TikTok to tell the product team to make them searchable. There's things you can do like filters and everything, but yeah, it's when there's 20,000 comments, you can't go through all of them. I try to cultivate the community, so I do go through them as much as I can in my free time, and try to boost the positive comments, like them, comment back, community building. But I do get exposed to the negative Nancy's and such, and so that can be hard.
Yeah. So we are on a podcast, and this is mostly an audio medium, but if you wouldn't mind, let's try to walk through in some detail what your most popular videos are trying to do here, because there's lots of conversations about people feeling some kind of way about the vaccine, and also retroactively feeling some kind of way about previous vaccines. And there's concerns that in the future, maybe we're going to stop vaccinating people for polio or for rubella and all the things that we all got as kids because of confusion around the science. Could you walk me through the creation of one of your assays and what you're actually doing? Because when I do the research, I open a lot of tabs to articles. When you do the research, you open up actually peer-reviewed journals, and then you do work in your lab. What are you doing when you are measuring your body's reaction to the vaccine?
Sure. I just want to, caveat, this is typically not allowed in most institutions. I'm lucky because I'm CEO. And
what is not allowed specifically?
Sort of experimenting on yourself, so to speak. Of course, Barry Marshall won the Nobel Prize for doing that, and ulcers and the bacterial origin of ulcers. But obviously, there's committees, there's a lot of regulation. So this is just my attempt to convince myself, really, because in the spring of 2020, when these newish mRNA vaccines came out, I was also skeptical. I'd been familiar with the technology. I've seen it ever since I was an undergraduate, which is 20 years ago. But I was nervous about it. So, what did I do? I went to the lab and did something about it. And so I do a lot of immunoassays, which is the type of assay that I do on TikTok, which is these microplates. They have 96 wells in them. They can hold less than a milliliter, 300 milliliters basically, 300 microliters of liquid in each well. And they can basically capture antibodies from your blood serum. And so then you can do a chemical reaction and see how many antibodies you capture. So that's basically what I've been doing since March, when I got the first vaccines.
Okay. And for our friends that are listening, that word is assay, ASSAY,
assay.
For testing things to determine their ingredients, to determine their quality. And you have this array of how many by how many was like
96. I use 96. There's also 384, and there's even bigger ones.
So big grid of little wells, you called them, and you honestly were thinking, I'm going to get the vaccine, but I'm also nervous about it. You were legitimately like, you had feelings about it. You're like, I'm not just going to do this and be like, this is awesome. I'm going to prove that it's awesome.
Exactly. No, I did those TikToks back in the day, and I was pretty nervous. I texted my sister and said, I'm about to get the vaccine. If you don't hear from me, that's what happened. I literally did that. I could probably find that text. That was on Valentine's Day of 2020. Or 2021. Sorry. So I was nervous.
What do you say to, my wife is an emergency room nurse. And she was like, vaccines, yes, but this mRNA thing was new. So what do you say when people say it was rushed, but it also sits on the base of decades of vaccine research? What was rushed and what wasn't rushed?
Yeah, so what was rushed was the regulatory aspect, which is slow as heck. I developed a new brain cancer therapy out of my Oxford work and UCSF work, and the 10 years of work, and it's still just going through the first phase of clinical trials. So that's mainly what was rushed. Like I said, the technology's been around for multiple decades. They initially being tested as a cancer vaccine But cancer is actually a lot more complex than microbes. And so it didn't really catch on, but thankfully that technology existed, so that foundation of technology existed so that they could just sequence the virus when they had it isolated. And then in a day, they can literally design it. And so they had it within a week of sequencing the genome, and everything else was execution, logistics, manufacturing, and regulatory. So the science was not rushed at all.
Yeah, that's a really interesting thing, and that was a point I was trying to make to some friends and family, was that they sequenced it over a weekend in January of 2020, but that's not the part of that. The rushing is the pushing and the getting it out and putting it into arms, right? It's all of the supply chain stuff, but the technology is definitely is there. So your nerves were more like, wow, we really got this thing out the door fast kind of nerves. And you wanted to see what it did to your body. So when you say experimenting on yourself, you're drawing blood on yourself and you're doing what to that blood to measure its success?
Yes. So, the antibodies live in the serum of the blood, which is the liquid part of the blood. It's straw colored yellow. And so I put my blood in a centrifuge and I can separate the blood cells from the serum, and I collect the serum. And I use that serum in my assays in order to do so. But serum has a ton of antibodies in it, and so you need to dilute it before you use it. And that's what I do in my TikToks.
Mm-hmm. And how do we know that the antibodies are antibodies for COVID and not antibodies for some other random stuff? How are you separating things and counting them?
Exactly. So the assay that I'm using is called an ELISA. We don't have to get into the acronym, but it's an immunoassay. It's called a sandwich ELISA, actually. So there's a bread. For the people watching video, you can see my hands, but there's bread, there's the meat, and then there's another piece of bread on top of that. The bottom piece of bread is actually the virus spike protein itself. So I buy recombinant, which means manufactured in a laboratory, in a manufacturing plant, through biotechnology. I buy recombinant spike protein. I coat the bottom of the wells with that. And then, so that's the bottom bread. Then I place my serum, which is diluted appropriately. That's the meat. Now, if there's any antibodies against spike protein, they will attach. And then the last piece of bread is actually an antibody that can detect my antibodies. And then it also has a marker on it, which gives us the color. So that's how it works.
Interesting. Yeah, that recombinant word is familiar to me as a diabetic because I use insulin that is made in a lab from recombinant DNA. So those capture antibodies are pre-coded onto that plate, and then you sandwich in the samples, right?
So it's not capture antibodies on the bottom plate. It's the actual antigen. The actual spike protein is on the bottom of the plate. Mm-hmm. And then my sample, which is a serum with my antibodies in it, and then an antibody against that for detection.
Okay. And then for folks that will watch the YouTube, the TikToks rather, that I'll put into the show notes, we end up with this kind of interesting gradient across multiple axes within this grid of wells, and it kind of goes from clear to bright blue. But then some of them were yellow. Can you explain the relationship between those colors and what success looks like?
Right. So there's two things there. So first, for every assay, you need to sort of you need a baseline, you need a frame of reference, which is the standard curve. And the standard curve is a known concentration of antibodies. It's basically a dilution series of antibodies from nothing to whatever is your upper limit that you set for the assay. So that's why you see always on the left side of my plates, you see two wells that are dark at the top and clear at the bottom. So it goes from most concentrated to nothing. Mm-hmm. And so that's how you get frame of reference, because I know exactly how much antibody is in each well. Mm-hmm. And then you can basically graph that. And then the linear portion, this is getting a little complicated, but the linear portion of that graph, you can directly, correlate to the levels in my blood to find an actual amount of antibodies in my blood. But so they start out blue because that's the first reaction. It's a redox reaction. Again, the darker the blue, the more antibodies present. And then this is an ongoing reaction, right? Mm-hmm. We don't want to overdevelop because that can happen. And so we want to stop the reaction with some sort of strong acid, and I usually use sulfuric acid or carboxylic acid, and that stops the reaction. It also turns the color to yellow. but it is proportional to the amount of blue. And the spectrophotometer, the plate reader, can actually give the numerical value for basically absorbance, how blue or how yellow is the well. And that'll correlate to the amount of antibodies in the well.
So if you don't mind, let me see if I can paraphrase some of that to make sure that I understand from the layperson's perspective, because this is super interesting. So keeping in mind that I'm a computer person and a lot of our friends who are listening are computer people as well, if you gave me something that was clear or white and it gradiented all the way up to blue, and you said, all right, we're going to call blue 100 and we're going to call clear 0, and then everything in between, whether it could be a linear thing where like baby blue is 50, or it could be a curve thing where we go, no, baby blue is actually more like 75. You give a curve that describes that gradient, but that control you have, that brightest blue, that's the top part of your y-axis, right? And if you did the experiment and a bunch of stuff came out deep, deep blue, then you've probably got the wrong y-axis and you'd need to then adjust that y-axis. Is that a fair understanding so far?
Exactly. Mm-hmm.
Okay. And then you're starting a process. You're like, hey, look at the antibodies, go, go, go. And they're like, yeah, we're going to go. And they start marching out and they start the process. If you let that process go, you might end up with a whole assay full of dark blue because time went by and everyone got to work. You're going to go and do it for some fixed time period and then stop it. And you're going to freeze time. You're going to pause the movie at a regular time. And then you're like, okay, look how active they were within this time frame. And then we do that consistently. And we know that like more antibodies jumped in, like the guys in 300 that were all like, let's go. And that's important. That's stopping of that reaction. Does that mean that people with fewer antibodies will eventually get there? Or is the idea that death will catch you before you get there?
No, yeah. I mean, they'll all advance proportionally. So.
Mm-hmm. Okay. So when we say that we have more, we get these numbers in the news like 400 times more, 1,000 times more. Those are big numbers. I mean, I don't think that the regular Joes and Janes, myself included, really understand what like an order of magnitude, much less multiple orders of magnitude means in the context of the biology of the body. If you have 1,000 times more antibodies, we tend as lay people to go and say, well, that must mean it's 1,000 times more effective, but it doesn't really directly correlate like that, does it?
No. And just to be clear, we're talking about, so in my tests, we're at 400 or 500%, not 4 or 500 times, which would be
so
2 orders of magnitude greater. Yeah. So it's only 4 times. So it's not a full order of magnitude, but it is significant because, as we know, back earlier last year, the Pfizer vaccine was 95% effective at 6 months with those levels. And so, how much higher can you get than 95? So it is sort of diminishing returns, but I think in the real world, depending, while all things equal, it could make a difference between someone being in the hospital or not.
Hey friends, there's something new from our sponsor Text Control. Their new product, DS Server, provides document services out of the box for all platforms and languages. Whether you want to integrate document creation, editing, sharing, or collaboration into your web app, DS Server provides the back-end technology to integrate that professional document processing. For example, using DS Server, you could integrate an MS Word compatible document editor into pure JavaScript, Angular, or an ASP.NET Core application. You can create PDF documents using Web API calls or even request electronic signatures from end users. DS Server is also hosted on-premise in your infrastructure or with your cloud provider, such as Microsoft Azure. And you can test DS Server without downloading anything. Create your first DS Server application in minutes by requesting a trial token on their dedicated website at dsserver.io. That's DSServer.io.
In your recent video, you had a couple weeks after and a couple months after, and it kind of like starts to decay. And is that the body forgetting or not caring? Why does the body forget?
Yeah, so that's normal. And people who have had COVID and have antibodies, this sort of natural infection, this natural immunity, their antibodies also wane because if the body is smart, it doesn't waste resources on things that it doesn't need to. And so if you're not exposed to that particular virus again, then the antibody settles down. Hot Vic Krishna has great TikToks on this, that actor in New York.
Yeah, he's lovely.
Yeah, so I recommend seeing those. I always point people to that. But basically, if you don't have an active infection, if you're not actively exposed, the body doesn't really need that protection. And so it sort of winds it down. Although we're in a pandemic, so we don't really want it to go down. So we need to boost and bring those levels back up.
Interesting. How come, though, we have things like polio vaccines or rubella vaccines that we take once when we're 5 and we don't think about again, but no one's walking around constantly exposed to polio?
Yeah, that's multifactorial. That's a great question. That's multifactorial. Number one, there isn't a lot of it around right now because of vaccines. So it's sort of chicken egg. But also, those viruses tend not to. They all have different means of mutation, and it just like think about the flu. The flu, we have to get a new shot every year because it's different every year. Because not only does it mutate, it recombines. So there's multiple different subunits, and it literally makes new combinations like a transformer, like a Voltron, basically. And so we never know what we're going to get. Thankfully, COVID hasn't really displayed that. There are mutations, as we're saying, with all the variants, but and that's why we can still use the original vaccine against the variants. Although, again, effectiveness is starting to sort of wane a little bit.
Mm-hmm. In your most recent video, getting your booster, you were surprised that you kind of shot off the y-axis and you had to do a little bit of adjustment there. It seemed to me like, maybe I'm reading into your video, but you seemed to me genuinely impressed with the immune response with that booster. What is it about that reminder 5 or 6 months later that caused the body to go, oh you're serious? I mean, I knew you were serious on the second shot, but I guess you really, really mean it. You really mean it. And the body just reacts. Why does that happen after 6 months?
Yeah. So again, Hot Vic Christian has a great TikTok video on this. But essentially it comes down to memory B cells. So, the vaccine is proven to not only produce antibodies, but also produces B cells and T cells that are targeted against, COVID spike, and, the body remembers. And so if you're exposed to the same, antigen or bug multiple times, the body realizes, okay, this is a problem, and ramps up its response. Now, back a year ago, when we were just starting to get the vaccine, we thought that the secondary immune response, that sort of jump, would occur between the first dose and the second dose. And it did. My first video showed that, and it was really impressive, but it really surprised me in September or October, or whenever I did, November is when I did the first booster, I think. It surprised me that it was like a multiple fold, 400% increase. My best, if I was betting, I would have guessed it might have been slightly higher than after the second dose, but not 4 times the amount, because it's a crazy amount of antibodies.
Yeah, it is. It makes me think about how the human body thinks about memory. And when you do staged studying and you're studying something, if you try to cram before the test, and then you go in the next day, you might do okay, right? But then it's probably going to leak out of your brain the next day. But if you do it in stages and you study and then you sleep and then you study and then you give it a week or 2 and then you study again, it moves from short term to medium term to long term. So it feels like these memory B cells at the beginning of long term memory, that true gaining of immunity that could last for months or years. Is that a fair analogy?
Exactly. And we need both, right? People saying, oh, it's not all about antibody numbers. It's both though, right? Yes, people who have had COVID and have some immunity will respond by creating more antibodies because they'll have memory. But it's better to have antibodies already at a high level to try to prevent that. So it's both. We need the B cells, the B cell memory, as well as the antibodies on board.
Is there room in the body? This is a dumb question on purpose, but is there room to keep track of all this stuff? There's so many things, so many bacteria, so many viruses. Are we confusing anything by, are we going to run out of memory? Cellular memory?
I mean, it's a great question. I guess it's hard to answer, other than, a lot of people are getting the booster and the flu shot at the same time. So think of that, number one. And also, if, you travel to some exotic foreign country, you're getting lots of vaccinations at the same time, as well as in the Army and in the service. And so at least from that perspective, no. But, the amount of antibodies in the blood is really, really high. And of course, the body sort of optimizes that based on exposure.
Mm-hmm. So how outside of your expertise is this? When went to Oxford, you've done postdoctoral work. When I see as a layperson someone in the lab doing the kind of stuff that you're doing, what specialization is this within the context of medicine, right? I don't think that my my GP, my regular general practitioner, either has done that since school or can do that kind of stuff. So help me understand the difference between MD, PhD, lab work, postdoc, and research.
Sure. I mean, it's all medical research. And of course, medical doctors, if they received the training, if they did a postdoc, maybe after going to medical school, may have done these types of things. It just happens to be my expertise. I've always done these types of immunoassays ever since I was an undergraduate, actually.
Oh, wow.
And so that's sort of my
You've
bias.
Been doing this for 20 plus years, 25 years.
Yes. I did undergraduate in Seattle, University of Washington, go Huskies. Great mentors there in neurosurgery, which is what I was originally intending to go into. And they really encouraged me to do the MD PhD because they were all very academic. They all published papers. They were some of the few neurosurgeons in the world who had NIH funding, RL1s, from the NIH and NINDS. So that really inspired me. And so I got the MD PhD in neuroscience. I did a lot of these types of assays, during that. And then, me being a total neuro nerd, I wanted to be the best at what I was doing. And so I postponed residency training, went to Oxford and did a postdoc. That's where I made my discovery that led to the drug that I developed. And then if that weren't enough, I did a second postdoc at UCSF, to launch the company. So it depends on your experience, but these are pretty basic techniques. Mm-hmm. If you have basic lab experience, you could probably run one of these assays just like a recipe in your cooking, because it's just steps. You just have to measure this and incubate that. So it's much like a recipe.
Was it challenging to go somewhere, like you said, publicly on the internet, whether it be TikTok or Instagram, and you've been doing this work at the graduate level for 22 plus years, and then, someone with a default avatar is like, yeah, well, says you, man. Doesn't that kind of feel like the death of expertise on the planet?
It's hard to hear for sure. So let's start there. Mm-hmm. And I think this is a great point to bring up, because I don't know if you watched the D'Amelio show on Hulu, not to plug that, but so I can sympathize with that. I'm not at that stratosphere, of course. She's over 100 million followers, Dixie and Charlie, but they really struggle with mental health, and the negative comments seep in, and they did for me too, because after that first video that really went super viral, 15 million back in July. And most of the comments being negative, I took a step back for a month. I was like, I don't know if I want to do this anymore. And so it does seep in, but then I also realize, the positive platform it's given me. I can, help fight misinformation, and you see real world impact, because that's literally what my entire career has been about. I've changed my career so many times, from neurosurgery to biotech and everything else. And now I'm a creator, but the real world impact. So it's all about happiness and impact for me. Having someone write in the comments, you are the reason I got vaccinated, or you're the reason I got boosted. And there's dozens of those. I haven't seen them all. It gets me every time. I mean, it's amazing.
Yeah. Some of the great comments I've seen going through your videos are like, this is what doing your own research looks like. And that's very inspiring. I've shown your videos to family and friends, and they were like, wow, I knew it was good. I knew it was working, but it's so nice to just see that blue in the corner and say, wow, look. And he was impressed too. And then when you go and you put it into your machine that takes the color and puts a number on it and then chart it, it's pretty hard to make those charts lie.
Exactly. I mean, and I was, I was, completely honest. My levels did wane by 5 months below 50%. And I was a little bit disappointed, and I made that video. I put it out there. If it went poorly, I would have probably done the same thing and documented it, and then said, we need to do better.
Yeah. Are you going to keep doing this? And maybe what's your time frame? Like every 3 months, every 6 months? Because now we're wondering, I'm assuming as a lay person that every year I'll just get a flu shot COVID combo, because I already get, as a diabetic, I get a flu shot every November anyway. Mm-hmm. What's your, not holding you to it, but what's your general sense of how you think things are going to go?
I think that's eventually where we're going to get to, a yearly combined flu coronavirus vaccine. And, lots of people are actually working on it right now and have been working on it for over a year. I don't think we're going to get there quite yet, because we're still in the middle of the pandemic. Things are still evolving. The virus is still evolving. Mm-hmm. And so, we're not going to be able to do that. So we're sort of, going to probably need another booster in 2022 that may or may not be modified. Who knows? It really depends on how things go. With this Omicron variant going wild everywhere, it's hard to tell. So, this is all real time science, which is why it's pretty cool. So, we just have to respond accordingly.
I want to make sure that we point folks to your primary research, which is your goal, your fundamental goal in life appears to be really building solutions to defeat brain cancer. I've got a friend right now who is battling glioblastoma, so this is very close to my heart. And you said that you discovered something and invented a thing that's now currently in trials to hopefully give people some relief.
That's correct. Yeah. So ever since I was in my undergrad phase, pre-med phase, I was always interested in brain cancer. It just fascinated me because there's nothing you could really do about it. Back then, that was, in the late 90s, there were literally 0 FDA approved drugs for brain cancer. And so that's what's really inspired me. So I've been working on it for 20 years at Oxford, along with some of my previous experience. I made a discovery, which led to me leaving my training in neurosurgery in order to start a biotech company of all things. So I can get this drug to patients because, I could probably impact more people with that drug rather than my own 2 hands. And so thankfully, we were able to raise lots of money, 8 figures, and get this idea I had through manufacturing, through the early regulatory. Now it's in phase 1 clinical trial at Moffitt Cancer Center for glioblastoma. And it's amazing. I still can't believe it's happening. And we've enrolled several patients. And so still waiting to see what's happening there. But yeah, it's inspired me to now, try to help patients now through my nonprofit, Cure Glioblastoma. And so that's where I'm doing all this vaccine work as well, because actually, I'm taking sort of a startup strategy through the nonprofit, and we're actually developing new technologies, doing our own research. And we happened to do a little COVID project last year because it was the pandemic, because we have a brand new antibody development platform, if you will. And so we did make some antibodies against COVID. And so this is actually the work I'm doing with my blood serum is actually really relevant to what we're already doing at the nonprofit. But eventually this could be a tool that could, develop antibodies for any disease. We're focused on brain cancer and COVID at the moment.
And you've also got your organization Brazen at brazen.bio, where you say real CEOs wear lab coats, where you're trying to make a connection between founders and creators who need curated resources to go and do things like drug development and product development.
Exactly. So, there's a founder-led movement in tech 10 years ago, obviously with the Zuckerberg et al. Now we're starting to see that in biotech, and we want to be part of the solution. Beyond that, we're also just trying to inspire people to consider STEM careers as well as communicate science on Instagram and TikTok and other platforms. And actually, Brazen Bio is signing creators who do STEM content. So we're giving them equity to do what they're already doing.
Have you seen any folks also commenting in your STEM work that you've inspired them to get involved in STEM or maybe switch careers and think that maybe pre-med's for them?
Yeah. So in my Instagram DMs for the past 7 years, there's not to toot my own home, but there's a lot of it, and I still can't believe it. It's amazing to see. And so now we have a Discord server to help everyone as well, so that everyone the users can help each other as well.
Fantastic. Well, I'll make sure to put links to all of your great organizations in the show notes so that folks can get involved and get into those Discords. Thank you so much, Dr. Shawn Carbonell, for chatting with me today.
Appreciate it.
This has been another episode of Hansel Minutes, and we'll see you again next week.